Hello all. So today I'm going to talk a little bit about background information regarding Cytochrome C and the main technique that we used in the experiment to obtain our data. So first of all, a bit about the structure of Cytochrome C. It's a monomeric protein, and the folding can be considered as a two-state equilibrium between the folded and unfolded states. The protein contains a heme complex, which consists of iron coordinated to four nitrogen atoms. This can be seen in the image below, where the heme complex is the gray molecules and the iron is the orange sphere in the middle. The heme is covalently bound to the protein by two cysteine residues. These form thioether bonds perpendicular to the plane of the heme cofactor, which gives the overall geometry around the iron octahedral shape. Another key feature of the protein is that it's very positively charged at physiological pH. This will play a big role in later on when we start to discuss how the changing the pH affected the stability of the cytochrome C. So now just to look a little bit at the function of cytochrome C in the body. It's responsible for transferring electrons from complex 3 to complex 4 during the electron transport chain. During the process, the iron in the heme is reduced from Fe3 plus to Fe2 plus, which is as a result of picking up that electron. The electrons transferred throughout the electron transport chain, and specifically in this process, serve two main purposes. The transfer of the electrons provide energy to pump protons across the inner mitochondrial membrane. This helps to create a proton gradient across the membrane, which is essential in ATP generation. Another thing they do at complex 4 is they reduce oxygen to water, and the reduction of oxygen is the driving force for all the reduction processes earlier in the electron transport chain. And this is the final step of picking up those electrons and really just providing the potential for all the other processes to occur. And it allows the electron transport chain to flow consistently and all the way through. So now a little bit of details about FRET, which was the technique that we used to collect our data in the experiment. FRET's a fluorescent technique that provides data on the distance between designated areas of a molecule. During FRET, the donor atom is excited by light, and in the diagram below, you can see the green circle is our donor atom. And then the energy released by the donor atom can either be transferred to an acceptor, which is the red circle, and this is a non-radiative fashion, or it is possible for the energy released from the donor atom to be emitted as fluorescence. In cytochrome C specifically, the tryptophan 59 residue is our donor molecule, and it is excited between 280 and 295 nanometers. The heme group in the cytochrome C is the acceptor molecule and has the potential to quench the energy emitted by that tryptophan 59 when it's been excited. This was possible this technique allowed us to uncover a little bit about the folding of the protein, because the more unfolded the protein was, the further apart the heme and the tryptophan residues were. And this increased distance meant that less energy was being absorbed by the heme. And so the fluorescence signal increased as the distance between the tryptophan and the heme increased. And this meant that we could track the unfolding of the protein, because the larger the fluorescence signal, the further apart the heme and the tryptophan were. And so in theory, the more unfolded they were. Thank you for listening, and I'll hand it off now to the rest of the group.